So today I will be presenting some of the work that we've done in using Campylobacter. I will preface it with stating that I won't be presenting any salmonella data just due to our time constraints, but I'm happy to speak with anyone, offline regarding some of those results. Okay, so this study focuses on the inactivation of Campylobacter in an in vitro system and also on the actual meat product. And what we did was we combined it with some organic acids, namely lactic acid and citric acid, and also acetic acid with Yucca. So just as a brief background, I'd like to give a little bit of information about Campylobacter. We know that it is a gram-negative motile rod-shaped bacterium, as seen in the picture there. We know that the strains are microaerophilic, of course, meaning that oxygen is toxic to this particular organism. And you can see the makeup of the gases that are used that are conducive for the growth of this particular organism. It is an enteric organism that is isolated in high numbers from feces of animals and birds. Of course, with it being an enteric organism, it affects the intestines. This particular organism, as it relates to the pathogenesis, is highly dependent upon the ability of the organism to penetrate the epithelial cells. And of course, we understand that the epithelial cells actually line the intestines. We all know the importance of the intestines and the function of the body that it has for our body. So if the organism is able to colonize in the intestines through penetration of those cells, that is where the process of pathogenesis starts. For this particular organism, the effective dose is considerably low, about 500 cells that are known to cause illness. In some cases, there are some documented cases where less cells have caused illness. Also, this particular organism produces a toxin, and this toxin in turn causes damage to the cells, inflammation in the body, and also that fluid loss. And of course, that fluid loss is what leads to the dehydration. The dehydration is what lands individuals in the hospital. The symptoms of Campylobacteriosis are very similar to that of other pathogenic organisms. Your diarrhea, abdominal cramps, vomiting, which many people do not go to the doctor for. So we do know that there is some underreporting as it relates to this particular organism. Okay, so this is just a figure that shows data from the CDC between 2000 and 2017. Through these years, you can see where poultry lies right here in this 41 cases. With some of the unidentified foods, poultry was suspected, just wasn't confirmed. So that number of 41 is likely higher. And again, that just goes back to that underreporting that happens. Okay, so let's switch gears just a little bit and talk about the problem at hand. So we know that organic acids are used throughout the food industry to control microorganisms, pathogenic organisms in different food systems, and of course, for the benefit of this presentation, poultry. We know that treatments such as lactic acid and acetic acid are being used. There's a lot of studies out there that have been published that show this use. These two have been, highly studied in the science world. But it's important to note that it has been documented that because poultry has some fat, we know that if the product is contaminated, those bacterial pathogenic organisms that we don't want in our food systems that fat can provide protection for those microorganisms. And when we think about acetic acid and lactic acid, citric acid, those are all diluted in water. And we know that poultry, by nature of the product, the skin is hydrophobic in nature. So when you dilute in lactic acid, citric acid, and when you dilute those in water, you can have a wettability issue there. Okay, I got a little ahead of myself. So that's basically what this particular slide says. So the whole idea is to create an environment where we can make sure that the acids that we are using to control these microorganisms on poultry, that they are actually able to make contact with all parts of the product. So my husband is a soil physics person, and he always creates these soils that he treats with chemicals to make them hydrophobic. So he treats them, and then he puts, beads of water on them, and then you can see the water beading up on the soil. So if you can just imagine, not necessarily to the extent of that, but that kind of gives you an idea of what these acids can do when they're diluted in water. Okay, so the proposed solution for this particular work is that we are proposing the use of a processing aid, which we are looking at these food-grade surfactants, because what they do is that they would break that surface tension that is created because of that hydrophobic nature of the skin to allow the acid to actually have the contact needed to further decrease the bacterial load should the pathogen be present. So Yucca is one that we explored for this particular study. Now I'll present some of the results that we have. Of course, we're going to build off of these results to do bigger studies on a larger scale because this was a benchtop study. So Yucca is what we use. It has FDA GRAS status, and it's also approved for use as an ingredient in food systems. It's used a lot in the beverage industry. Now, Yucca is desirable because we do know that there are surfactants out there, such as sodium lauryl sulfate and others that are, used and that are popular. But as you all are aware, we're moving into this trend where consumers are demanding these more natural, environmental friendly products, and so the food industry is scrambling to continue to meet that demand. So Yucca is able to fit that definition. Okay, and this is just a picture of the plant. It comes in different sizes, shapes, but this is just one. It's native to the southwestern United States and Mexico, and it does have those surfactant capabilities. Okay, so that brings me to the objective of the study. The objective of the study was to investigate the ability of these organic acids, lactic acid, citric acid, and acetic acid, in combination with this Yucca extract to control Campylobacter on chicken skin. Okay, so just to give you a brief background on what we did to prepare for the study, we used 5 different strains of Campylobacter. They were cultured separately in Mueller-Hinton broth, of course, which is a common broth that is used for culturing of this organism. 2 consecutive transfers were done to further purify our cells. And after that, the cells were harvested by centrifugation. The pellet was resuspended in 0.85% saline, sodium chloride saline, to attain about 9 logs of the organism. And then after that, we pooled those 5 strains together to form a cocktail. Now, I'll mention here that we use the 9 logs, of course. If you are a poultry facility and you have 9 logs of an organism, you have a big problem, right? You have a big problem, and you have to address or answer some serious questions. But of course, in the world of research, we inoculate at these high levels so that we can very clearly see inhibition. So that is why those counts are so high. And it was this cell suspension that was actually used to inoculate in both the broth study and in also the study where the product was used, the poultry product was used. Okay, so as it relates to the preparation of the treatment solutions, you can see we had the lactic citric acid at 1.5% and then at 2.5%. And then we had the acetic acid at 1% and at 2%. Now, for both of these acids, we did filter them, sterilize them by filter sterilization. And you can see the size of the pore and all that information there. Also, for the Yucca, we looked at the Yucca alone, which is the YEX here, Yucca Extract. We looked at it alone and also in combination with the other acids. So the Yucca alone was at 0.15% and then 0.3%. And then when we combined it with the other acids, it was at 0.25% and 0.5%. Okay, so moving into the inoculation of the treatment solutions and the chicken. So the first bullet point basically refers to what we refer to as a time kill assay. So this is where we inoculated tubes in which the treatments were present. So this is in the absence of a food product. And this is very important, of course, in food microbiology, because if we see no effect in the absence of the food product, then we really have to consider whether it's worth moving into using a food product. And because funds are limited, we have to ask those questions ahead of time, okay? So that first bullet point basically refers to what we did. We had 20 mL of each treatment solution that we evaluated in the large sterile Pyrex tubes, and we inoculated it with 0.2 ml of the Clostridium. I'm sorry, thinking Clostridium. I have a student that's working on Clostridium now. But Campylobacter, the 0.2 ml of the suspension was inoculated into each of those tubes. And we sampled at different time periods, 30 seconds, 60 seconds, and 90 seconds. And in that last bullet point, that is the bullet point, that is the point where we used the chicken skin. We inoculated it with 50 microliters of the cell suspension. And then after that, we took the spreader and we made sure that the inoculant was spread over the sample. After that, the inoculated sample was held for about 1 hour to allow for bacterial attachment to the product. Okay, so as it relates to the chemical treatment of the chicken, we had the solutions, 200 mL of each solution that were in sterilized Pyrex beakers. And we basically took the product and dipped it into the treatment there. You can see that on the screen there. And we did use sterile thongs to transfer the skins into the treatment solution and also from the treatment solution to the stomacher bag. Okay, so here our immersion treatments, they were in there for 2 minutes, and after that we evaluated the product for survivors. Other skin samples, another set was also immersed for 2 minutes in the treatment and then stored at 5 degrees Celsius because we wanted to evaluate the effects of time as well. And after that, they were subject to microreal analysis, just as that product or those samples that were simply treated and then analyzed. And then you can see during the storage, they were at 5 degrees Celsius in these sterile stomacher bags. Okay, so as it relates to the microreal analysis, the first bullet point relates to the time kill assay that was performed. At 30, 60, and 90 seconds is when we evaluated those, and they were plated out after those time points directly from the tubes. And then, of course, for those samples that where we actually used the product that actually went into the stomacher bag containing the 100 mL of peptone, they were stomached using a laboratory stomacher to remove any survivors from the product into the diluent. So we used those filtered stomacher bags where the product is on one side, and then of course you have the diluent there. And so the diluent is what we sampled. Okay, so all samples were plated on MH Mueller-Hinton agar, supplemented with the Campylobacter, the Preston Campylobacter selective supplement. They were incubated under microaerophilic conditions at 42 degrees Celsius, and colonies were counted after 48 hours. And the same was done for those samples that were stored at 5 degrees Celsius for 24 hours. Okay, so very quickly as far as the statistical analysis, viable counts of Campylobacter were transformed to log value. Analysis of variance was carried out using the SAS software. 0.05 was used as the significant levels, and we did perform 3 replicate experiments for each experiment. Okay, so as it relates to the results, you can see our controls were our saline, and you can see in this, of course, this table represents the results for the time kill assay. So you can see there's a column for 30 seconds, 60 seconds, and 90 seconds. So we have our saline control there. We have the next 2 are our Yucca extracts at our different concentrations of 0.15 and 0.30. And if you just look across there, you can see where the reduction lies at 30 seconds, 60 seconds, and at 90 seconds. So very little reduction for those for the Yucca alone. Next, we move to the lactic citric acid. You can see the 2 concentrations that were evaluated there, the 1.5 and the 2.5%. And you can see of those 2, the 2.5% at 90 seconds yielded a 2.66 reduction. Okay, so then we move to the lactic citric acid samples in which we combine the Yucca. Okay, so you can see there the 1.5% for the lactic citric acid, and then the 2.5% for the same acid as well, in combination with the 2 different concentrations of the Yucca that was used. And you can see of those, of course, the last one that's highlighted there in the box there, you can see the lactic citric acid at 2.5% plus the Yucca at 0.5% exhibited the greatest inhibition at 90 seconds. Okay, so this is the same for acetic acid. You can see the data is presented the same. We have the saline as our control, and then we have the Yucca at the 2 different concentrations, the acetic acid at the 2 concentrations. Of course, the acetic acid at the 2% yielded a 2.74 reduction at 90 seconds. And then you can see for the acetic acid plus the Yucca, the same trend as with the acid previously discussed, the 2 different concentrations of the acid and the 2 different concentrations of the Yucca. And then you can see the last one, the acetic acid at 2% and the Yucca at 0.5% yielded 3.02 log reduction at 90 seconds. So I can go back. On this slide right here, the 2 that you see in the red boxes, and then on this slide, the 2 that you see in the red boxes, that is what is represented here, because those are the 2 treatments that moved forward when we added in the product to the study. So you can see here, this is presented, of course, also as log reduction. The control samples there, the distilled water and the saline. And then you can see the lactic citric acid at the 2.5%, and then in combination with the Yucca at the 0.5%. And then you can see it goes down to the acetic acid with the concentration by itself and also with the Yucca. And so you can see here the log reduction. If you look at, for example, if you look at the right here, where we have the lactic citric acid at the 2.5% plus the Yucca at 0.05%. 2-minute immersion, you can see the log reduction was at 2.24, and then you can see when we did the immersion plus the 24-hour refrigeration at 5 degrees Celsius, you can see the reduction is at 3.8. So if we look down at the acetic acid, you can see at the 2.0%, 2 minutes, you see a 2.72 reduction. And then when you add that 2 minutes plus the 5 degrees Celsius, it being held at that temperature over 24 hours, you can see there is a 3.92 reduction. But you can see with the acetic acid, it's a little bit higher, 2 minutes, the 2.88, acetic acid plus the Yucca, excuse me. At 2 minutes, the 2.88 reduction, and then at the 2 minute immersion time, plus it being held for 24 hours at the 5 degrees Celsius, you can see the 4.14 reduction. Okay, so in conclusion, the lactic citric acid at the 2.5% or at the 2.0% with the added Yucca exhibited the largest reduction in populations in vitro, just in the tubes. It shouldn't say on the chicken skin there, it should just say in the tubes. But then where we have the addition of the second bullet point, when we add it in to the product, you can see the Yucca and the lactic citric acid and the effect that it had on the product as well. Okay, so just very briefly, I would just like to acknowledge the support of the U.S. Egg and Poultry Association for this particular study, and also our graduate students both at Alabama A&M University and at Iowa State University. We had students kind of working together on these studies. Okay. 1 minute to spare.